I hope you enjoyed it. And we're going to get started here in the CHS room. We have Dr. Brian Gress from UC Davis. He came up from California for us. He's going to talk to us about spotted wing Drosophila resistance management in California.
All right, thanks for coming, everyone, and thanks to the organizers for putting this together. Okay. Thanks for coming, everyone, and I'd just like to thank the organizers for putting this together and making everything run so smoothly. Okay, is this better? Okay. Thanks, everyone, for coming, and thanks to the organizers for putting this together and making it run so smoothly. Today, I'm going to talk about some work that we've been doing in California over the past year or so, looking at spinosad resistance in spotted wing Drosophila. I'm also going to talk about some possible resistance management techniques that can be implemented, but also some unique challenges to resistance management facing the system. So first, I just want to give a brief overview of the invasion of this pest into California. So it was first found in commercial strawberry fields in Watsonville, California in 2008. And at the time, it wasn't clear, there was no species ID, so it wasn't clear exactly what growers and researchers were working with. By later that fall, high densities of this pest continued to develop in these fields. And by later that fall, very high population densities were observed. In spring of 2009, we saw a mass infestation of cherries. And by fall of that year, there was also high levels of infestation in caneberries and some in strawberries in the coastal regions as well. At this time, it was eventually ID'd as being Drosophila suzukii, which allowed researchers to get some more information about the biology and natural history of this pest. There was an available literature, but it was from Japan, and so it needed to be translated. But one of these studies from Kanzawa in 1936 reported that Drosophila suzukii was first reported as a pest of cherries in Japan as early as 1916, which is consistent with what we were seeing in California. After its arrival in California in 2008, it quickly spread throughout the rest of the country, as you can see from this map. So the colors here correspond to the year where it first showed up in each state. So after its arrival in California, it quickly spread through the Pacific Northwest, popped up in Florida, and by 2011, it had spread throughout the entirety of the eastern portion of the US. 10 years after its arrival, this is what the current distribution looks like. It's widespread throughout the US, also in British Columbia, Nova Scotia, Ontario, and it's also become a problem throughout Europe as well. So it's rapidly becoming one of the most severe global pests of berry and other small fruit production. So I'm not sure if it's really not there or if it just hasn't been documented. So that's kind of unclear. Yeah. It could easily have something to do with both the heat and the dryness of the pest and their preferences. There are also a number of unique challenges facing growers in terms of trying to manage this pest and some unique characteristics that make it a particularly damaging insect. So first, of course, is its preference for ripening and otherwise marketable fruit. And so it differs from other Drosophilids and vinegar flies in this respect, which typically prefer overripe or rotting fruit. And females are able to infest underripe and firm fruit using this unique egg laying structure, which has saw-like teeth on it, which they can use to cut into the fruit, deposit their eggs, and that, of course, leads to larval infestation. It also has a very fast life cycle and overlapping generations, which when combined with its high reproductive capacity, means that populations can rapidly expand and get out of control. The adults are also highly mobile, so as we have seen it spread throughout the country, it can quickly take over new areas and move to new regions. Additionally, this pest has an extremely wide range of both crop and non-crop hosts. So currently more than 130 plants have been identified that this species can develop on from across 31 different plant families. Additionally, it's been documented that it can develop off of bird droppings and cow dung. So basically, it's next to impossible to prevent populations from building up in these non-crop habitats, and then of course moving into the crop setting where it causes mass infestation. So what can be done about it? One of the most important things is sanitation, meaning try to reduce breeding sites, and this entails removing and disposing of mature and overripe fruit and eliminating any alternative habitat like called fruit piles or abandoned fields, since these provide a source for Drosophila to build up populations, and these ultimately sustain infestation. However, sanitation isn't enough, and growers are still rely very heavily on insecticides for controlling this pest and preventing infestation. So this figure here shows a summary of efficacy data that was collected across 10 different states and a number of different crops. And basically what it shows is a number of different products that have been used against SWD, and ranks them as ranging from having anywhere from no activity against this pest to having excellent activity. And what you can take home from this is basically that conventional growers have a number of good options, including pyrethroids, organophosphates, and spinosins that together form a highly effective spray rotation program that can prevent infestation and adequately control this pest under most conditions. But organic growers are much more limited in terms of their spray options. Currently, Entrust or spinosad is the only organic insecticide with high efficacy against this pest, which means that it's both critical for the organic production of these crops, but also by not having an effective spray rotation partner, that increases the likelihood of resistance emerging. In California, we've been doing most of our work in the Watsonville area, which is located in Monterey Bay region of California. And this is a major hub of berry production in the US with high concentrations of raspberries, blackberries, and strawberries, both conventional and organic. And so in all of these plots, SWD are being exposed to spinosin-based sprays. So just for reference, there were approximately 1,100 kilos of spinosins applied to about 12,000 hectares of berry crops in this relatively small region in 2016. And perhaps not surprisingly, there have been growing concerns over recent years that spinosad seems to be losing efficacy against SWD. And so because of this, we decided to participate in a national resistance monitoring survey in 2017. And this survey was conducted as part of a USDA Specialty Crops Research Initiative grant. 8 states from throughout the US participated, and each of these states used the same bioassay protocol to test the flies so that results would be directly comparable across these different regions. And the bioassay protocol that we used was developed by Rufus Isaac's lab at Michigan State University and Ash Sael's lab at the University of Georgia. And it's a simple bioassay that ultimately entails adding 1 milliliter of your insecticide solution to one of these 20 milliliter glass scintillation vials, shaking it around to make sure that the inside is coated, emptying out the excess material, and then leaving that vials to dry overnight. The next morning, we would then add 5 male and 5 female SWD, leave them for 6 hours, and then perform our mortality assessment where we classified each fly as either living or dead. And we tested the flies at 2 concentrations. So the first was the LC50 concentration, which should kill 50% of susceptible flies. And the other concentration was the LC99 times 2 concentration. So this is the concentration that should kill 100% of susceptible flies. So if you have any survival at this 99 times 2 concentration, it raises an immediate red flag that you may have a problem on your hands and indicates that you have some more work to do. So since we were particularly interested in the Watsonville region, this is where we focused most of our sampling attention. And so we sampled from 5 different sites throughout the Watsonville area, all of which came from commercial caneberry plots. We also sampled from an additional location in Winters, California, and specifically these flies came from USDA Wolfskill Germplasm Repository, which is an untreated mixed fruit experimental orchard. And since it's untreated, the intent was that this would serve as a susceptible baseline or a point of comparison with our Watsonville flies. And so here are the results from that monitoring study. So this figure here shows the proportion of flies surviving at each of our 2 concentrations. So the blue bars show survival at the LC50 and the orange bars show survival at the 99 times 2. The first 5 sites are our Watsonville sites, and then the last site is our untreated wolfskill population. And so starting with that bar on the far right, our wolfskill population, we see pretty much what you would expect from a susceptible population. We saw about 57–58% survival at that LC50 concentration, and we saw 0% survival at the 99 times 2 concentration. However, looking across our Watsonville flies, we see survival ranging anywhere from about 73% to 90% at that LC50 concentration, and between 2 and 17% at the 99 times 2. So this was kind of confirmation that the reports that we've been hearing from growers did seem to be true. It seemed like we were seeing a reduction in susceptibility of these flies to this particular material. And so we wanted to understand the full extent of the problem and also understand what's the potential for this problem to get worse. So we performed a resistance risk assessment study where we exposed spotted wing from Watsonville to high concentrations of spinosad to see if we could push resistance or select for resistance to increase it in the population. And so we started this by initially screening about 700 Watsonville flies at that 99 times 2 concentration. And this gave us about 80 survivors. Using these flies, we collected eggs, reared those progeny to adulthood, and then tested those flies again and repeated that process for 5 generations. At that point, we performed dose response analysis where we tested flies against a range of spinosad concentrations so that we could calculate LC50 values of our own for each of our 3 strains, including the untreated wolfskill population, the Watsonville population, and the Watsonville population after 5 generations of selection. And just go back quick. For this work, we performed 2 sets of mortality assessments, one at 6 hours and one at 8 hours of exposure. And the reason for this was simply that at this time we were still trying to optimize this bioassay protocol. But they do give slightly different results, and so I'm going to present both of them just for the sake of completeness. So starting with the 6-hour time point, here's what we found. So the figure here shows concentration of spinosad on the x-axis and mortality on the y, and this table here shows the relevant values from this analysis. The first column shows the LC50 values for each of our populations. So with that untreated wolfskill population, we find that concentration of about 46 parts per million kills 50% of the population, whereas the Watsonville population had an LC50 value of about 355 parts per million. And you can calculate a resistance ratio from these by dividing the wolfskill or the Watsonville LC50 by the wolfskill. And this gives you a resistance ratio of 7.7, indicating that the Watsonville flies have about an 8-fold increase in resistance over the untreated wolfskill population. However, when we compare the Watsonville flies to a Michigan population that was tested by Rufus Isaac's lab, we see a resistance ratio of about 22, so much higher yet. And this is especially concerning. I do want to stress a bit of caution with this because these studies were performed in different labs. It's possible that small differences in environmental conditions or handling of the flies could account for some of this disparity. But nevertheless, both of these values are quite concerning. How about in our selection line? So after 5 generations of selection on the Watsonville population, we see the LC50 increase from about 355 parts per million to 424 parts per million. And this population is now about 9 times more resistant than the untreated wolf scale site. Looking at the 8-hour time point, we see a reduction in the LC50 values as you would expect because we've extended the duration of our bioassay. So now we find that the wolf scale population has an LC50 value of about 29 parts per million, while the Watsonville site comes in at about 153. And we also see a slight reduction in the resistance ratio. So the Watsonville flies have about a 5-fold increase in resistance over the wolf scale site and about a 12-fold increase over that Michigan site that we compared to previously. And again, after selection, we see the LC50 increase in the Watsonville population from 153 parts per million to about 228 parts per million. And so what we can conclude from this is that spinosad resistance is emerging in the Watsonville population of SWD, and that the continued use of spinosad does have the potential to push resistance beyond its current levels. However, having said that, I do want to point out that the response to selection that we saw in this study was relatively low compared to similar types of risk assessment studies that have been performed in other species. And so this means that at least in the near term, there's some reason to be optimistic that spinosad will maintain at least decent efficacy against spotted wing. But in the longer term, this poses a real threat to organic production in this area, particularly because growers don't have any real alternative to turn to. So again, that's another challenge for resistance management as well. So that stresses the need for additional work to continue to understand this problem and to ultimately develop new control strategies and management tools, as well as better monitoring tools as well, so that as soon as we identify that there's a problem or try to identify any potential problem early so that growers can respond and take action immediately. So what are some, so with this in mind, we started a series of experiments this past growing season to try to better understand this problem. And the first that I want to talk about right now was a study where we were trying to understand population dynamics of resistance through the Watsonville area. And specifically, we were interested in understanding the spatial distribution of this, of resistance. So understanding how widespread this is through the Watsonville area, but also interested in understanding temporal patterns of resistance. So is there a seasonality to resistance? Are there fluctuations in resistance throughout the year? And specifically, we predicted that throughout the growing season, as growers are applying their sprays, that resistance would likely continue to build and increase throughout the growing season. But then during the off season, once those sprays stopped, we predicted that resistance may decline once those sprays stop. And the reason that we expected them to perhaps decline was for a couple of reasons. So one, at the end of the year, growers remove the hoops that they grow the caneberries under. And that would provide an opportunity for the resistant flies underneath those hoops to disperse and mix with more susceptible flies in the surrounding vegetation that maybe haven't been exposed to the insecticides. And the second reason is that oftentimes there are fitness or survival costs associated with resistance. Physiologically, resistance isn't free for these organisms. And so once those sprays have stopped and spinosad isn't prevalent in the population, those resistant flies may be at disadvantage that would ultimately, you would expect, lead to lower frequency of the resistant flies. And so to test this, we continued sampling through our Watsonville populations, except this time we focused on 3 time points throughout the year. So an early season time point where we collected flies in June, which coincided with when many growers were beginning to apply their first sprays of the year. We had a mid-season time point, which was in August, and a late season time point that was in October. And then for each of these time points, we again performed our dose response bioassays so that we could calculate LC50 values and then track changes in resistance throughout the season. And so here's what we found, starting with the early season data. Tables are set up similar to how you saw before, with LC50 values for each of our sites, and then resistance ratios giving an indication of how much more resistant the Watsonville site is than the, the susceptible wolf seal population. And so just with a quick glance here, oh, and I should point out that all of these were done at the mortality assessments were performed at the 8-hour time point. So what we see here are resistance ratios that range anywhere from 5.6 to 9.3, all of which are higher than the resistance ratio that we found at the end of 2017. So this essentially rejects our hypothesis that resistance would decline during the off season and that the early season time point would be when the flies were most susceptible, which of course isn't what we expected, isn't what we wanted to see. As for the mid-season time point, we see similar pattern where here our resistance ratios range anywhere from 4.3 to 7.3. So it might look like that's lower than the early season time point, but that's not statistically significant. Likely just noise associated with the sampling of flies in the field and the bioassay itself. And currently we don't have data on or complete data set for the late season time point, but the preliminary results look like they'll fall roughly within the same range of about a 4-fold increase in resistance to about an 8 or 9-fold increase in resistance. So what this means is that resistance appears to be stable in the population through Watsonville. And so the potential for a strategic spray program that targets susceptible flies, like at the early portion of the year, the usefulness of this may be very limited. So what options are available to growers who may have resistance in their fields? Well, I want to kind of shift gears a little bit now and talk about some options that growers could implement to reduce their reliance on insecticides, as well as increase the efficacy of materials that are already available. And I'm going to briefly talk about some areas where there's some preliminary data that looks promising and where additional research is needed in the future, but also some data that came from an organic research and extension initiative grant that we've been participating in California for a couple of years now. And I think that ultimately this should give you an idea of the different types of approaches that can be integrated and what a resistance management program might ultimately look like. First, I want to start with the chemical control side of things. So our focus, at least for the time being, has been on trying to increase the efficacy of the spinosad to bring it back to those pre-resistance levels. And so one area that's received a lot of attention over the years has been the use of feeding stimulants, particularly sugar. And there's been a lot of mixed data that I've seen coming from studies in different regions where they've tried to use sugar as a feeding stimulant. In some cases, people have found it to be very effective and to increase efficacy. In other cases, people have found absolutely no effect whatsoever. And yet other cases, people have found positive effects, but have decided that it's not worth it for them because it clogs their sprayers or it leaves their field sticky, and it's just not worth it. But one thing that's become clear is that there's been no standardized concentration of sugar used across these different studies. And so the results aren't necessarily comparable anyway. And I know going forward into this coming year, there's going to be some folks who are going to try to solve this problem and do some field trials where they do standardize the amount of sugar and try to find an optimal middle ground where maybe you can use a concentration that's high enough where you do see that boost in efficacy, but not so high that you're clogging sprayers and leaving your fields all messy. A similar approach that has been gaining some attention recently is also the use of erythritol, which is a non-nutritive natural sweetener. It's not been used so much as a feeding stimulant, but more just looking at its activity against spotted wing by itself. And there are a number of folks who have been looking at this recently, including Jana Lee at the USDA in Corvallis, Oregon. I saw her give a talk on this a couple of weeks ago, and they're claiming to have identified that this material by itself is enough to cause physiological disruptions in spotted wing and actually create water imbalances that make females incapable of laying eggs, and that they've seen some benefits of this in some field trials. The other area that I want to spend a bit more time talking about is trying to identify synergistic rotation partners that can be paired with spinosad to again increase its efficacy. And one product in particular that we've been interested in California is JetAg, which is a sanitizer or fungicide. It doesn't have any activity against spotted wing by itself. So why are we interested in it? Well, it's because of some work that was done by Phil Fanning at Michigan State University. So he ran a laboratory dose response bioassay with spinosad similar to those that we've been doing, except he had 3 different treatments. So he had spinosad by itself, spinosad in sugar, and spinosad in yeast mixtures. And both the spinosad in sugar and spinosad in yeast treatments were intended to, again, test this idea that to test both of these materials as feeding stimulants to see whether you could get a boost in efficacy. And what he found was that both the spinosad and sugar treatments had very good efficacy, just like you would expect. But when mixed with yeast, he saw a complete drop-off in the efficacy of spinosad. We don't know why this is the case, but we do know that there are yeasts present on the exterior of the fruit, on the skin of the fruit. And so it's possible that by using some of these sterilants to come through and clean off these yeast and get rid of them, that you could see a boost in efficacy of spinosad as well. So that's one possible benefit. The other possible benefits associated with this product are that we know spotted wing are highly attracted to the yeasts that are on the skin of fruit. So by removing them, you might also make the fruit less attractive. And I know Alan Schreiber has done some work and has some data supporting this, showing that spinosad rotations with JetAg are more effective than rotations with Grandivo, for example. Another possible idea that's been raised is that if flies consume this material, that it could clear out the gut microbiome of the flies. So it may not kill them immediately, but it could have a number of sublethal effects on these flies, because we know that in Drosophila, the microbiome is important for, it affects lifespan, it affects the organism's ability to fight off disease, as well as reproduction. It's important for reproduction as well. So there are a number of kind of promising, or a number of things that make this a particularly promising option to rotate with spinosad. Okay, so next up, I want to talk about some work that we've been doing with using cultural controls to try to manipulate the microclimate of the canopy of different crops and try to focus on some behavioral control aspects. And I'm also going to talk about a new paper that was just published by Von Walton's lab at Oregon State University, where they just published a very exciting new behavioral control as well. And so first, the study that we participated in, this again was part of the National Organic Grant that we've been participating in. So this was a pruning study, and the idea for this came about based on some work from Diepenbrop and Burrick in 2016, where they found that the interior portion of the canopy in caneberries had much higher levels of infestation than the exterior portion of the canopy. And of course, the interior portion differs in that it's more shaded, it's more humid, and it's cooler. And so the thought is that this is just a more preferred environment for females to be laying their eggs, as opposed to the exterior portion that has direct exposure to the sunlight. And so the question was, by pruning the canopy and thinning it out and increasing light exposure, can you manipulate the microclimate in a way that deters females from coming in and laying eggs in the first place? And so in California, we worked in raspberries, but this was replicated throughout the country on blackberries and blueberries as well. And in each case, we had 3 different pruning treatments. So we had an unpruned control, which was our high canopy density treatment. We had, so in California on raspberries, our medium pruning treatment, we removed 25% of the canes. And in our high pruning treatment or low canopy density treatment, we removed 50% of the canes. And then we measured a number of different traits associated with productivity of the plants and infestation, including light penetration into the canopy, temperature, yield, and larval infestation. And so what we found was that pruning significantly impacted canopy density early in the season. So we found higher levels of light penetration early in the season, but by the end of the season, the canopy had filled in, and we didn't see those effects anymore. Because of that, we also found no impact of pruning on canopy temperature. And surprisingly, we found no impact of pruning on yield either. So even our treatments where we removed 50% of the canes had statistically the same yield as those that were unpruned. So this was pretty exciting. Also, we found that both of our pruning treatments had significantly lower larval infestation than our unpruned control. Here you can see that mean larvae per berry in our untreated control were around 0.4, while both of our pruning treatments had very low levels of larval infestation. So this is an option. I should just say that the results here weren't necessarily consistent across all of the regions or across all of the different crops that were tested. So for instance, Michigan and Minnesota found no impact of larval infestation in their trials, and a number of different sites found that, especially in the high pruning treatment, that they did see a reduction in yield associated with that. So this is something that can provide benefits, though it's not guaranteed depending on where you're at or what crop you're producing. But it is a good option that could have real benefits. And additionally, there's the added possibility that by thinning out the canopy, you could see increased spray coverage as well. And this is a question that I know Kelly Hamby at the University of Maryland is interested in, and I think pursued this past field season. So we might be seeing some data on that soon as well. The second behavioral control study that I want to talk about, as I said, was a study that was just published by Von Walton's lab at Oregon State. His lab has been working with a team of chemists and have developed this food grade gum material that has an attractive proprietary compound blend in it that is apparently highly attractive to SWD. So attractive, in fact, that it seems to distract the females from the fruit, and instead females preferentially oviposit, lay their eggs into this gum material. And field trials that they ran have found that the presence of this gum in the fields can by itself can reduce infestation between 50 and 76%. So this is brand new, kind of hot off the press data. But now that this has been published, I fully expect that in the coming year, more regions are going to be testing this across a number of different crops. And so this is certainly something that I'll be keeping my eye on, and that I find to be a really exciting avenue for future control. Another thing I just wanted to briefly talk about here, because it's my understanding that this has been becoming more common in production in Washington, but that's the use of exclusion netting to enclose the crops and to physically exclude SWD from getting in. There's been a lot of work coming out of Minnesota, a group in Minnesota, showing that this can be highly effective at preventing infestation. It has the obvious drawback of being very costly, but at least in some situations and in some crops, it might make sense. And there's even been work showing how high tunnels and other bird netting structures can be modified to include this exclusion netting to make it a bit easier. It's important that if you're going to go this route, that this netting is installed prior to SWD's arrival, because otherwise you're simply going to be trapping the flies within these tunnels. You're not going to be getting any of the benefits. Another concern with this is reduced pollinator access, which can be a real problem. And again, I know that people, I think in Michigan and Minnesota, have been doing some experimental releases of pollinators into the exclusion netting to see if they can combat some of this work. So I just wanted to touch on this because it is highly effective, but as I said, there are some obvious drawbacks associated with it, and it might not be plausible for widespread use. Okay, so the last area that I want to talk about is the use of biological controls. And so biological control, of course, is the use of natural enemies to target pests. There are a number of benefits associated with biological control, so they can provide long-term, cost-effective control and reduce reliance on insecticides. And there have been a number of surveys done throughout the US and Europe trying to find some natural enemies of SWD. And some work has shown that there's at least one fungal pathogen, as well as some predators like ants, spiders, rove beetles, and minute pirate bugs that will attack different life stages of SWD. And there's been some laboratory studies that have shown that suggested you could get high control using some of these. However, every time this has been taken out into the field, we found that it provides absolutely no control. So the focus now has switched to classical biological control, which is slightly different. It's the approach of identifying and releasing natural predators from the pest's native region. Obviously, there are some concerns about going to some place, collecting organisms, and then releasing them into a non-native habitat. So there's a very rigorous process that has to occur in order to get approval to do that. So there has to be very strong data showing that these have high target specificity to your pest, and that they're not going to be attacking other important native organisms. There's also some just practical concerns about whether or not these can be mass produced and released, and whether they will establish in the population. This work has been led by Kent Dane at UC Berkeley and Kim Holmer with the USDA in Delaware. And they've conducted a number of collecting excursions through China and South Korea to try to find parasitoid wasps that, again, are specialists on SWD and that will attack it. And so far, they've found 3 different parasitoids that from the native habitat that do attack spotted wing. However, only one of them has shown to be highly specialized on SWD. The others are more generalists and will attack other organisms. And so this one that is most promising is Ganaspis brasiliensis. And actually, they submitted a proposal to get this to do some experimental releases of this in the field. Their initial petition was rejected, but it had mostly positive reviews. There was, I think, mostly just one negative review that held it up, and they've been working to. So they have this organism in quarantine in the Fresno area of California currently, and they've been working on it in the lab, and they've been doing a number of studies in the quarantine facility to alleviate some of the concerns that the one reviewer of this petition had. And they plan to resubmit this in early 2019, and at least Kent Dane is very optimistic that they will get approval to do experimental releases of this. So this is another area that's, again, on the horizon, not here currently, but that could provide a major benefit and an alternative to chemical control options. And so with that, I would just like to thank everyone who has been involved in this work and funding sources. And with that, I would be happy to take any questions. Yep.
Commercial growers, actually the organic fields is kind of like a pest haven. You know, it's there's a lot of organic fields, and that's nearby some of the commercial fields. What is the life expectancy of an adult? I call it a doofus fly.
So that's kind of a tough question to answer what the life expectancy is. You can house them in the lab and they will live for months and months and months. What their life expectancy is in the field is a much more difficult question to get at, because obviously those aren't the same conditions. And with insects, it's not like a large mammal where you can tag them and track lifespan in the field. So it's actually very difficult to get mortality data on insects. So I don't have a good answer to that, unfortunately.
Just as an observation, I've got an early variety raspberry, and this been through the years has been tested, and I have very little problem with the doofus flies larvae in the berries. But as you go into the blueberries or your later variety blueberry, it gets to be a real pain in the rear. Because they just multiply as the season goes on. But it doesn't appear to show up in the very early fruit in this area anyway.
Yeah, that's absolutely true. That it does take a while for populations to build in most locations. However, in California, for example, in cherries, which come in the spring, we will see very high levels of infestation, but that likely is a different scenario because we have such mild winters. We're probably not having the population completely collapse over the winter months. So things are a bit different where we're at than places where it maybe gets colder during the winter and populations decline more. So yeah. Yep.
Okay, I have two questions. The first one is, given what you just said, how many times would a grower spray, for example, in those fields that you were testing for resistance? And when would be the start? and when would be the finish of the spray season?
Off the top of my head, I don't know exactly how many times they would spray, but I know that in some cases, sprays will start as early as late May and continue. So I did my last collection of the year this past year, late October, and they were still harvesting, and I assume still spraying at that time. So that's, basically from May through October, I would say.
So a little bit longer than what we experience up here.
Okay.
For a
What is
season,
what is it here?
It's usually, when do we start? End of June, and go until last spray would be early September.
Okay.
And so my second question was, you told us about Entrust. Are you testing resistance on any other products?
So we have been looking at malathion as well. So we initially did some monitoring work on Mustang Max, malathion, and Entrust. We found absolutely no evidence that there was any change in susceptibility to Mustang Max, and so we've stopped work on that. We have seen mixed data on malathion to the point where I'm not sure what's causing the variability in the data, or sometimes I'll run tests that look very concerning and then I'll run them again and they seem to be behaving as they should. So that's an area where we now are going to be focusing more attention. We did continue to monitor for malathion resistance through 2018 as well. Had some of those same issues, but it's an area that we want to now start investing more effort in, because if there's a problem with malathion, that's probably a bigger concern yet. And then the other thing that we haven't tested, but is kind of the obvious next step, would be to look at something like Delegate, which is very similar to Entrust. And so you might expect that to be kind of transferable in terms of the resistance.
Hi, back here.
Yep.
With your pruning trials, and then also the experiments where you've done small plots to compare different applications of products, they're not necessarily killing the fly. Concern I've had with that research is that you're basically giving a choice to the fly in the field as to whether or not it wants to go to the low-hanging fruit, as it were, versus a less suitable environment. Is there any discussion amongst you and your colleagues as to how to translate those sorts of trials to the field scale, where you would potentially be changing pruning strategy for the entire field?
It's a really good question. And it's not something that I had previously considered. And I'm not sure there would be a good way of getting those data other than just doing it at a larger scale, which of course, pruning is a labor intensive strategy by itself. And so it's I guess potentially a high risk, high reward sort of approach to take. But yeah, I just, off the top of my head, I can't think of any way to address that concern without simply doing the test and just doing large scale pruning. I do work with one organic grower in Watsonville who, before we even started this work, did high levels of pruning in his blackberries, and he swears by it, but that's very anecdotal and obviously not anything too convincing. So.
Hi again.
Yep.
Kind of partly, maybe as an answer to this lady, is that once that we see the appearance of the doofus fly, that you have to spray between 6 and 7 days. But even after that, as you go from the raspberries into the blueberries, even with your spray program that you have, after a while you lose control of the infestation. It shows tough. and the final statement of that is that also the wild here in Whatcom County, we got in Canada, we got a lot of wild blackberries. That's a lovely haven for
Yep.
doofus fly.
Yeah, the wild hosts are a real concern. As I said, it's obviously wild blackberry would probably be one of the worst, but there's no shortage of things in the environment that spotted wing can develop on. And I think that we don't have good data at this point to know whether it would be cost effective or time effective to actually go through and try to remove some of those. That might not be practical, but we do know that those wild hosts can be a source of SWD and a source of major infestation. So yeah, it seems like something should be done with them, but it's not clear what can really be done. Yep.
There we go. Is entrust and delegate single site mode of action products?
Yes, I believe so. Yep.
I don't know if this is a question or an observation that for the longest time as an old-time farmer that I thought that this doofus fly didn't really overwinter because of especially if it gets down good and cold in the wintertime.
Mm-hmm.
But I guess they pretty well hide in, even like the ladybugs in behind everything.
Yeah, so there has been on some of these national grants that we've been participating in a lot of effort to try to understand the overwintering. So this fly has a specialized winter morphology. So if you rear these under cold conditions or short day length, the flies actually emerge as adults. They're much darker and they have smaller body size and much larger wings. So there's a distinct phenotype that corresponds to flies that presumably makes them more cold hardy. And so they have special adaptations for overwintering. Now in really cold areas like upstate New York, Michigan, there are people there who still think that who think that it's not likely that these are capable of overwintering there. And there's an idea that at the beginning of the year they may be moving in from lower areas. That's a question that still hasn't been nailed down exactly. We don't know whether that's the case, but there's so Joanna Chu, who's at UC Davis, who's on the slide here. She's a molecular biologist, and she's been developing molecular markers to distinguish different populations of spotted wing from throughout the US to try to address this question, to see whether if you could basically track these flies using genetics to see whether each year the northern populations are being repopulated by flies from the south, or whether they're distinct populations that the flies are overwintering in. So yeah, it's a good question, but I think that unless it's very cold winter conditions, the flies have no problem surviving. Yep.
So a lot of our raspberries and blueberries here are mechanically harvested. And there's always some that make it past the harvester onto the ground. And my question is, do you think those could have any significant influence on SWD pressure throughout the season?
Yes, I do. Dozens of larvae can develop from a single rotting fruit. So yeah, I don't think it takes much in the way of, again, just fallen, rotting fruit or cull piles. It doesn't take much to produce potentially a large number of flies that then can provide a source of infestation. So to the extent that you can get rid of those, I think you'll see a benefit.
All right, I think that is it. Does anyone else? We probably have time for one more question, but it looks like we're wrapping up. Awesome. Well,
Right.
thanks,
you.
Dr. Gress. So we're wrapping up just a little early here. I'll have those pest credit sign-up sheets in just a second over where we've been signing up here by the entrance. Just a reminder, if you have a chance to review the session, you have a chance to win a $5 Woods Coffee gift card, and we really appreciate your responses. Just a note about these beautiful lanyards you have. If you're going to head out after this session, there's a drop barrel as you leave through the main entrance, so we'd love to have them back if you don't want them. And I think that is it. So yeah, I'll be over with pest credits in just 1 second.